One of the labs in the early 20th century that was examining the passing on, genetic inheritance. Um, but we're doing so and trying to go beyond Mendel, was the lab of Thomas Hunt Morgan. Morgan was an embryologist. Okay, an embryologist, they study the embryonic development of animals, plants. By studying the development, what happens in the different stages, you can begin to see also what genes must come into play and in what sequence. Now, Morgan based most of his work on studying the embryonic development of fruit flies. He wanted to see how things are passed on generation to generation and how this affected fetal development as well as adolescent, which we would think of as larvae, and sexual developed fruit flies. Now, going with fruit flies is actually a really cool thing for him. And he used this as what we refer to as a model organism. A model organism is an organism that, well, has traits that are great for your experimental design, but are still universal. Because you don't want to pick an organism to study and do developmental or cellular or genetic studies on, and it's so esoteric that your findings are only good and only apply towards that one organism. Fruit flies, they are an animal. They are an animal, eukaryotic, multicellular, have different stages of development, fetal, larval, things like that. But what's really good about them is they breed at a high rate. One male, one female, they will breed. They will be drawn to each other. They will breed. Um, just being in the presence of each other, as long as there's food, there's water, and they're healthy, they will breed. Not all insects are like that. Some insects are cyclic. They only mate, during certain months, during certain seasons. One and done for the year, and they're dead. Well, fruit flies, like I said, as long as there's food, there's water, they have a very short lifespan. They're only alive for a few weeks. So, soon as they're sexually mature, they're going at it. And, their generation time, less than 2 weeks. That means that fertilized egg gets laid, fetal development of the egg hatches, out comes the different larvae, which goes through different larval stages. Then you get, a non-sexually mature adult that will become a sexually mature adult. The sexually mature adults will begin breeding almost immediately. Next generation eggs get laid. All of that will occur in less than 2 weeks. And what's really awesome about these guys? 4 chromosomes. That's it. They only have 4 pairs, 8 total, which is much better than our 23 pairs, 46. Keep in mind, there are plants and animals out there that have more, some have less, but 4. And it's a eukaryote. This is excellent. Morgan's lab, his grad students, his undergrad workers, they were cataloging and characterizing the different traits that they could find in fruit flies. They found that there was some variability in different traits, what Mendel would call characters. He found that there was some variability, but there was things that were predominant throughout. These predominant or quote unquote normal appearances of the phenotype, he referred to as wild type. Any alternatives to the wild type, he referred to as mutant phenotypes. Great example is here above my head. Up at the top is the wild type eye color. Turns out fruit flies have the wild type, the normal eye color is a nice deep dark fire engine red. And I mentioned last chapter, eye color in fruit flies, turns out it's a pathway. It's not one gene, multiple alleles. Here you go, different alleles, different eye colors. No, it's a pathway. And different steps have different enzymes or different transport proteins. Each enzyme, each transport protein is the product of a different gene. What we have found is that the different steps, different genes responsible for going from no eye color to that nice deep dark fire engine red, that if you interrupt it at different steps, you end up with different eye colors. White, brown, cinnabar, sepia, just to name a few. The white, the brown, the sepia, the cinnabar, those are mutant phenotypes. Each is due to a mutation in a different gene. Other mutant phenotypes could be due to body color. Under normal conditions, the wild type body color is a tan with black stripes. A mutant phenotype for body color is all black. Wild type wing shape is wings that extend beyond the body of the fruit fly. Look above my head, you see the artist drawing of these fruit flies. A mutant phenotype is what they refer to as vestigial wings. These are little wing nublets that basically they just, basically have about as much flying power as me waving my fingers right now. The genes, wild type is the quote-unquote normal. Any alternative to that is referred to as a mutant phenotype. Now, Morgan and his lab, they were repeating Mendelian genetic studies. They went through, they studied, and, well, what they found is that even in fruit flies, Mendel's ideas, Mendel's theories held up predominantly. Take two true breeding for a certain phenotype, breed them together, as long as one's dominant, the other's recessive. Mate them. F1's all dominant, F2's 3-to-1 ratio. Repeat, repeat, repeat. Held up until they came across an exception. The exception to Mendel's rules, Mendel's laws, his ideas, happened to be an eye color. What they found when they crossed red-eyed with white eyes is, well, something that just doesn't hold true. Keep in mind, Mendel's work with pea plants. Pea plants are, for all intents and purposes, hermaphroditic. They have both the male and female parts, male making the pollen, female parts housing the ovum. They're all in the same flower. So, chances are the genes being passed on, the allele frequencies and different things like that are going to be the same whether you're talking pollen or egg. With fruit flies, distinct male, distinct female. When they did the red to white, and they did the study, they found that there was an anomaly. If they took, red-eyed males, crossed them with white-eyed females, everything followed Mendelian genetics perfectly. But when they did the opposite, white-eyed males, red-eyed females, things didn't work out quite right. When they step back and look at the numbers, okay, 1-to-1, white-eyed male, red-eyed female in the parentals, bred them, counted the F1s 2 weeks later, everybody's red eye. Allowed those to breed, counted the F2s 2 weeks later. 3 to 1, 75% of the F2 offspring were red eye, 25% were white eye. But when they looked a little closer, all the white eyes were male. This does not compute. For if it was truly Mendelian genetics, you should still see the 3 to 1, 75% red eye, 25% white eye. But that 25% white eye should be 50-50, equal distribution, male to female. But it wasn't. In the F2, all females were red eye. Only half the males were white eye. How do we explain this? Well, back and forth with different ideas until Morgan figured out that there is only one true difference between males and females in the animal kingdom. Our chromosome. That one chromosome difference. The sex chromosomes. For many of us animals, especially us mammals, XX versus XY. Yes, there are other versions out there in the insect kingdom and in the bird kingdom, in the animal kingdom, or in the avians. They may have, ZZ, ZW, things like that. But for fruit flies, they follow just like we mammals do, XX female, XY males, which led to the idea that the difference must be occurring on the X and Y chromosomes, because that's the only difference between males and females. Yes, a Y chromosome is why I'm sitting here looking at you, with the phenotype of male. For many of you listening to this, XX is why you look female, why you have female reproductive organs. The Y is why I have male reproductive organs. When you looked at just the phenotypes in this experiment, red eye female, white eye male, everybody in the F1 red. But right here's the problem. Mendelian genetics does not explain that. Until you enter into the 20th century and you start discussing chromosomes, working off of Sutton and Bolivar, or whatever his name is, who studied the sea urchins. Can't remember his name right now. Sorry, my apologies. Start equating things to these chromosomes. Still don't know what they are, still don't know how they function, just know it's proteins, sugars, some nitrogenous compounds. But right here, this is the difference. Yeah, it's drawn, looks like a shepherd's hook. Turns out that the Y chromosome in fruit flies actually does look like that. Morgan was able to figure out that the only difference between males and females is right there. All the other chromosomes are exactly the same. This applies to us mammals. Chromosomes 1 through 22 are pretty much the same between all of us. It's that XX or XY that's the big difference. So when they mapped it out, they put it on W plus. Remember I told you last couple of chapters, when it comes to genetics, look at how the nomenclature they're using. We're talking about a white-eyed mutation. So the gene responsible for that's going to be referred to as W. W plus means it's the wild type, means it's the working, the quote-unquote normal. W nothing means it's the mutant, giving you white eyes. Females have 2 copies. 2 X's, thus 2 copies. Males only have 1 X, thus only 1 copy. Take the parentals, breed them together. Here's the gametes for male, an X and a Y. The female will have 2 X's. Now they didn't draw it here, but the row is going to be just a repeat of this row that's shown. Fill it out. XX females, they are heterozygous. W plus W. W plus wins out because it's dominant. They're all red. Look at the males. Males are W plus nothing because the W gene's not carried on the Y. Set up the Punnett square for the F2s. Notice everybody here gets W plus. Everybody here gets the Y. Here's the major difference. W plus, W, W plus, W plus, homozygous red. W plus nothing means these are going to be red-eyed males because the only gene present is W plus. These heterozygous females, W plus W, just like we saw in the F1, W plus wins out because it's dominant. These are heterozygous dominant females. Here's our white-eyed though. W, nothing. There you go. Only way it could work. We now look at this as proof positive that Mendel's characters, his traits, have to be carried on chromosomes, have to be carried on specific chromosomes. It's only explanation on the 3-to-1 all male, 25% all male white eyes. Turns out W's carried on the Y chromosome. We've isolated the Y chromosomes, we've sequenced it, and yeah, we've been able to identify the W gene, and yes, almost a century ago, Morgan and his lab were correct.